presentations being given to the Department of Earth and Space Sciences seminar series on August 27th, 2020. And the title of my talk is Advancing Sustainability Through Scientific Collaboration. I want to start off by just acknowledging all of the folks that are co-authors on this paper. Many of these are students in, or former students of, the Environmental Science Graduate Program. And a number of these slides are generated from the direct result of a lot of effort from students in my former ecological methods class, which they participated in from 2010 through 2014. I'd also like to thank Cody Meshes for some of his help outside of class. He was not a student in that class. And then there's a number of colleagues that are from the College of Business, including Dr. AJ here and all of his students that participated in the grant that I'm going to talk about. And then funding was supplied by the EPA, Columbus State, Columbus Waterworks, and the Department of Earth and Space Sciences. Algal blooms are a problem. They're occurring more frequently. In a minute, I'll show you that it's a pretty widespread problem, but these algae are either single-celled organisms or multicellular organisms, and they can become extremely prolific at certain times under certain conditions. As you can see here, these boys wading through shallow waters that are absolutely covered in filamentous algae. The reason for this problem is that we are releasing tremendous amounts of nutrients into the water in the form of nitrogen and phosphorus. That stimulates the algae to grow. They are generally limited by the availability of nutrients in these aquatic systems or in the oceans. And when you add them, the phytoplankton start reproducing very effectively, and they develop what's called an algal bloom, which we just saw a picture of. Those algal blooms, either phytoplankton or filamentous algae, basically cover and intercept all the light that is coming into the ecosystem. And when they do so, they have lots of ramifications for all the organisms. And ultimately, when they die, the microbial community will break them down, and that will suck the oxygen out of the system because they are going through the process of respiration, which uses oxygen, and that leads to very low oxygen levels in our lakes and some of our bays and parts of the ocean, and that can create fish kills and all sorts of other problems. And it also leads to massive amounts of deposition of decaying organic material on the bottom of our lakes and streams and oceans. Global phenomenon, we see this is some older data from 2008, but what I like about this graphic is that it's showing in green all the areas that are lightly impacted by humans, and then as those colors get closer to black, we're seeing colors that indicate really heavy impact of humans. And you can see in the areas where it's red, like in Europe and Eastern North America, you see lots and lots of dead zones or other zones that have very, very low oxygen caused by human activities. We're seeing this expansion in this coastal zones where we're seeing very low oxygen, and that has very significant impacts on fish recruitment, fish growth, production, because most of the production occurs in these sort of continental margins. And low oxygen, of course, stresses the fish and all the other invertebrates and things in these ecosystems and can fundamentally change how our oceans work. We mentioned this a global phenomenon. One of the solutions might be to actually use algae to solve algal problems. And so that's what I'm going to talk about today, how we might implement the use of algae, which grow like crazy, take up nutrients out of water, which is why they're causing problems in coastal zones, but we might actually be able to use them before the water ever gets to coastal zones to solve this problem by removing the nutrients before they get there. Okay, so essentially what we typically have is a wastewater treatment facility, which is a potential source of lots of nutrients into natural water bodies, which flow to the oceans. And the way that typically works is you'll have the wastewater treatment plant is essentially doing a couple things. They're removing the solids, and then they're also trying to remove the nutrients, which are generally soluble. What we can do is we could actually capture that water or a portion of that water before it goes out to the river and drive it through an algal treatment system so that the algae have a chance to take it up inside a contained environment. And if we do that, then they will capture the nutrients inside the algae themselves before we let that water go back out to the river. So this is a way to clean up that water before it could be discharged. Nutrients don't just come from wastewater treatment facilities, but many wastewater treatment facilities in the United States do not have any kind of nutrient controls. And there are some that do, but not all of them do. Many of them do not. Those solids that are collected also have nutrients and things, and they're usually treated in many different ways, but one of the more common ways these days with wastewater treatment plants is that they have a biodigester. And so they put this stuff in there, and anaerobic digestion occurs with microbial communities breaking down those solids. And when they do that, they release CO2 and various types of burnable gases, including methane. And then we can take that methane gas, clean it up a little bit, and create electricity, which we can use for powering things. So in a sense, a wastewater treatment plant actually can generate not all, but much of its power based on this biodigestion. So just collecting the solids and doing that. But they also need to deal with these nutrients that are being emitted into our rivers, which flow to the oceans. So one way to do that, of course, with the algal treatment system, and we could improve the production of those algal treatment systems by using carbon dioxide from the biodigester. So we can even close this loop in a sense by taking a waste product of the biodigestion, which is CO2, and we can infuse that into these systems and try to improve the overall production of algae. I'll talk about that in just a minute. So what we did to start to study this kind of interaction is we developed a partnership with the Columbus Waterworks. This is a picture of their south plant that's on the Chattahoochee River. So you can see the Chattahoochee River. Here's Alabama, and this is the south plant here. These big circles here are not crop circles. That's the place where the water is clarified before it is released into the Chattahoochee River, and you see that here. This picture here is of my 2010 ecological methods class that first started to develop original ideas for an algal treatment system that we could use for doing experiments. And these are some more pictures of those students in 2010 developing the head tanks, and we ended up doing some additional construction and remodeling of the design in 2011. So you see students working on that there. In 2010, at the end of the semester, some of the students were interested in this project enough that they decided that they wanted to choose to do a grant writing exercise. So they wrote a grant, and then with some editing help from me and some other information, we put together a grant that we submitted to the EPA P3, which is People, Prosperity, and the Planet. It's a student design competition, and we were rejected. So we didn't get the funding, but we at least had a draft of some of our ideas down on paper. During this whole process, we were constantly making changes to our algal treatment system. We wanted to have lots of different replications. So you see each of these troughs is a miniature algal treatment system. So algae would grow as you flow water over it. And you can see we had a design where the water flowed and just went right back to the plant. We had some designs like this one where it actually recirculated into a tank, and then that tank recirculated the water. And then our final iteration of the system is shown here, where we have actually two tanks in here, and so these two systems operate independently of one another, but they're recirculating the water. I'll show that in just a minute, if this works. So here's a picture of the finalized design that we used, and this is important because I'm going to talk about some experiments we did in this design. Let me just walk you through it. I'm not going to do the video thing for the moment, just save a little time. So these are the head tanks. We bring water from this gigantic brown thing up here. That's one of the clarifiers you saw in the picture, the little circle things. We pump water down into these tanks here, and then that water flows down through these troughs, and in those troughs there are tiles that the algae can grow on. They can grow attached to these areas, and as the water flows over those tiles, the algae will pick up the nutrients, but the algae remain attached to the tiles, and we can harvest the tiles. Water goes down into these big containers underneath this, which you cannot see. Then they're pumped via pump back up this pipe back to each of these. So this is a separate unit here, and there's a separate unit here. These giant tanks you see here are full of CO2, and we use that to test this question about whether or not injecting CO2 into this system improves the production of algae. So in the grant that we submitted in 2013, we changed things up a little bit. We had an environmental science portion, but we also had a business team. They were looking at the financial feasibility of doing something like this, and we were looking at this question of, does CO2 improve performance of these algal treatment systems? And the results of those experiments were pretty clear. We found that so this is a plot of the algae collected from these different flowways. So here's a set of flowways where no CO2 was infused, just air. And this is a set of flowways where you see algae growing that were infused with CO2. Notice a nice green carpet there. And what we found is that by day 14, so these are dates, not actual day of the experiment. The experiment started on the 10th, and by the 14th day of the experiment, in these green infused areas, we saw a lot more volatile solids or biomass, an estimate of biomass of the algae. So there's a lot more algae that we were able to measure than compared to the controls. So these are means and standard deviations you see plotted here. And in fact, that same result was supported by another measure, an independent measure of the amount of biomass in these systems, which is called chlorophyll A. Chlorophyll A is a light capturing pigment. It's what makes plants green. If you have more green pigment, that means you have more algae in the system. And so what you see here is that no difference on the first 7 days, so they pretty much behave the same. But once those systems got lots of algae, we saw an increase in the production of chlorophyll A. And so we had more biomass in the treatments that were infused with carbon dioxide. So if you want to stimulate these systems, all we have to do is pump in a little CO2. We looked at nutrients as well. So this is a plot from so this is the very first day of the experiment. Here's the last day of the experiment. And you can see the solid line shown here with the open boxes. Those are the controls. So no CO2 added. We get a slightly higher end point, but along the line, they're pretty much doing the same thing in terms of the nitrogen content. So the water, this is the amount of nitrogen in the water itself. So we're starting with very dirty water, right? Lots of nitrogen. By the end, we see a lot less nitrogen, okay? But if we add CO2, we found very low levels of nitrogen in the system. So by adding CO2, we got a lot more algae, and we reduced the amount of nitrogen in the system. It didn't matter for the phosphorus measurements. It didn't matter whether they were in control or treatment. We saw very significant declines in the total phosphorus. So that suggests to you that the treatment process that we were doing was efficient at growing algae and reducing nutrients, which was our goal. So we took these results and we went to Alexandria, Virginia, and we competed in the EPA National Design Competition. And so this is our team here on the right. This is the other team on the left, as I'm looking at the screen. And you see the team from Johns Hopkins. Now, the reason I included Johns Hopkins is because when we went to present our results, we were saying to everybody, look, we're growing algae, and this algae can remove nutrients. The exact opposite was happening in Johns Hopkins. They were growing some algae, but basically they were taking algae that somebody else had grown, and they were digesting it in a biodigester. That's what this thing is here, and they were making methane gas from it. So I struck up a conversation with this particular gentleman here, Pablo, and we decided that it would be really great if we could collaborate together. So we worked up all our samples. We sent him a bunch of samples from our experiments, and we decided to start a collaboration. So just to make sure everybody's clear on how this might work, this is not a picture of our schematic, but it represents the sorts of things that we were doing. We have wastewater treatment plants that have liquid and solid manure that they liquefy, and they put into a tank, and then that goes into a digester, right? This digester is a sealed container where there's no oxygen, just a bunch of microbial organisms. And then we can feed into that our algae, right? So this is represented by the renewable material. And what we were interested in knowing is if you put algae and sewage sludge together, can you make more energy? Can you get more gas and so forth? Now, the way that we approach this, or the reason we approached it this way is because we knew that most of the wastewater treatment plants already had biodigesters. They were already making methane gas. And so this was a way for them to be able to take and add an algal treatment system and then have a sustainable way to deal with that algae that they produce. So they can generate even, we wanted to know, could they generate more production, more gas, more biofuel? So what we decided to do were three basic things. We decided that we were going to analyze the composition of the algae. We needed to know who's in the algae, what are the biochemical composition of the algae, because some types of algae can be consumed by microbes and some not so much. We also wanted to characterize how much biogas they can actually produce. How much methane can you get when you do things that people typically report, which is pre-treatments I'll talk about in a minute, or whether they want to digest it with the human sludge. And then finally, once we have those results, we wanted to plug those results into a model and say, does this even make sense? I mean, even if it works, does it make any sense from an energetic perspective? Just to illustrate that point, imagine for a second that you wanted to make biofuels from corn, right? So you've all heard about corn ethanol, right? Well, the analyses that have been done have suggested that we get very little additional energy from that process than the amount of energy you have to put into it. So it turns out it does yield energy, a net amount of energy, but not very much. It's a pretty small proportion. So that suggests it's really not energetically efficient to grow corn to make energy, but it might be energetically efficient to grow algae for energy. So that's what we're going to do in these next couple slides. So I'm going to show you the results of some of the work that Dr. Bahutsky did and his team, right? He had a whole team of folks. This is a table, and you're never going to be able to read it, particularly since it's not projected, but you don't have to read it because I gave you a little cheat sheet here, right? The results of this analysis of the biochemistry of these algae suggested that they're mostly carbohydrates, right? They're sugars and starches and things of various types. Complex carbohydrates, but about 45% of their mass is carbohydrates. The rest is proteins and fats, right? So those are the three main components of these living organisms. And so that would suggest that there's at least some energy there, right? Because you know, if you eat carbohydrates and you digest them, you can get energy from those carbohydrates. If you break down proteins, you can get some energy. You get a little bit less. Lipids have tremendous amount of energy, but the lipid levels were only about 20% or less in these algae. So not too many lipids in this type of algae. The other big point from this analysis is that if you look at all the potential calories that could be utilized by the microbes to make methane gas. So what they're doing is they're taking that carbon molecules and they're making them into methane. If you think about that process, there's a theoretical maximum yield you can have based on the amount of carbon that's in the system. And that turns out to be way higher than what we actually measured in the lab. So what we found is the microbes could only digest about 57 to 65% of the theoretical maximum yield. So hopefully that makes sense to everyone. So in other words, if I gave you a candy bar and the candy bar had 100 calories in it, if you ate it, it would be like saying you can only get the energy from 65 of those calories instead of all 100 of the calories. So we fed them 100 calories of algae, but they couldn't utilize all that to make essentially 100 calories of methane. We also then took these algal isolates, so these algal treatment samples, and we ran them under a series, I say we, Pablo's team ran them under a series of different treatments. Here's the control, right? We've got to have a control. We don't do anything to them. This is just the algae put into the system. And then we also did a bunch of different pre-treatments. And it doesn't really matter all these, but these are basically ways to try to chop up, mash, degrade the algae a little bit so that makes it easier for the microbes to digest the food. In the same way that you'll put food in a blender so that you can have a smoothie and it's easier to digest, well, we did the same sort of thing, but we didn't do it physically. We used thermal treatment, so we heated it just like you would cook food. We also used acid on it. We used alkalis on it. We did all combinations, okay? And if you look at the results, you see that methane production, which is shown along the y-axis here, actually increases relative to the control in all of these treatments. So if you heat it, preheat it, if you add these other treatments, you get more methane. But notice it's nowhere close to the theoretical maximum, which was over 500. So even if you pre-treat it, that didn't do much. But it's important to do that because you get more, look, you can get significant increase in the methane production, 30, 40%. So it can be important. It's not 30 or 40%, but it's reasonable, right? So you do see some increase. But what about that question about what if we mix it with sludge? What if we mix the algae with the sludge, throw it back in the digester, what happens? So this is the same kind of slide. Right here, we're looking at the control, right? So this is our control, and we're comparing that control, which is 100% algae, to different mixes of algae to sludge. So here's 90% algae and only 10% sludge, just a little bit of sludge, all the way up to no algae and 100% sludge here. And if you look at it, all of these are significantly different from the control. So if you add any bit of sludge to these samples, you're going to get more, you're going to get a total increase in the production. And we control, by the way, for the volume. So we're making sure we have the same amounts of material, carbon-based per gram material. So we're not just like adding more stuff to the mix. We're actually reducing, we keep that constant in these experiments. So if we look at the amount of methane production, it goes up quite a bit, and it's not at 500, but it's very, very high. We're up near 400. So we're seeing a pretty significant increase in the overall production if we mix in some of that sewage sludge into the mix. Remember I said that what we were really interested in at the end of the day was finding out whether this makes energetic sense. Should you ever do this just from an energetic? Does it really make energetic sense? Do you yield more energy doing all this stuff? Or would you just be better off taking your algae out and just letting it degrade as compost or something? Well, we didn't test the compost, but what we could do is we could take all the data that we got from these earlier experiments I just showed you, and we could plug it into a model. This model was trying to estimate, if we pretended for a second, we could scale up to a full-size wastewater treatment plant, full-size biodigester, the size of this room, gigantic biodigester. Would it be energetically smart to do this? And the way that we quantified that decision was whether or not the energy that you get out of it is greater basically than the energy you put into it. So we use this ratio of energy output to energy input, or the net energy return, okay? So if that number is above 1, that means that it's energetically beneficial to do something, right? If it's really highly above 1, then it's really smart to do something. So these graphs I'm showing you are that estimate, like is it smart or not to do this based on the model predictions? And then this axis right here is showing you a time frame. So biodigestion does not happen instantaneously. You put the goo into this container and it takes time for the microbes to work on it. And the longer you take to work on it, obviously, the more it digests what's in there. And so you see that they peak out the energy. The algae are getting, or the algae are being completely destroyed by the microbes, and then it just kind of stops. No more additional benefit. Okay. Now, what I'm showing in this graph is the heat portion of what we're doing. So we generate some heat as a result of this process. We also can generate a little bit of electricity from that methane, right? We can use that. And then here's what the one we really care about. So let's just talk about the total energy. Each of these lines represents a different treatment. So the first one to look at is the control. This is just algae. So if we look at just algae and we put it into this model, it suggests that, hey, it is beneficial to digest algae, right? The net energy return is above 1, which is good, right? In fact, it's almost 3. So that's good. But notice all the other pre-treatments we did, regardless of whether we heated it or whether we added, even though we got more methane, energetically, it doesn't make sense to do it because it takes energy to make those chemicals or to raise that heat. So at the end of the day, you'd be better off just sticking the algae right into the biodigester and not pre-treating it at all. Even though you'll get less methane, energetically, it makes more sense. So we can do this exact same measurement with the sewage sludge. So in here, we've got, again, our 3 different graphs of heat, electricity, and total. We'll just talk about total. Again, along the y-axis, we're just plotting the energy, the amount that we get out relative to the amount we put in. And again, if it's above 1, it's good. But here's the thing. The control in this one is just algae. That's that black line. Notice that now we've flipped everything. The black line is at the bottom. The reason the black line is at the bottom is because every single treatment makes more sense than just doing the algae alone. So all of a sudden we had this eureka moment when we did these experiments. It's like, wow, it looks like if we combine algae with sewage sludge, we can actually not only make more methane, but we can do it in a way that energetically makes a lot of sense. It's more beneficial from an ecosystem perspective. And what's really interesting about this, and the thing that probably sealed the deal for why we got this published so quickly, was that if you look at this little star right here, so this particular treatment right here, in the model, that is the highest return of all of them, right? It has the highest return. That is a combination of about 10% algae and 90% of sewage sludge. Well, if you think back to that wastewater treatment plant, they're going to mostly be putting sewage sludge in there, right? We're only going to be putting a small amount of algae into that system. So if you're putting in roughly about 10%, you're going to actually maximize the efficiency of the entire system, which is perfect. So these results, which were all based on models and observed data in the lab, suggest that if we do this at scale, it's going to not only work, but it's going to work really, really well. So what are the take-home messages? One, if you infuse wastewater treatment, algal treatment systems with carbon dioxide, you're going to increase the biomass of algae, which is good. And it's about 40% increase, by the way, right? 2, that there's not that much of an effect of CO2 on the nutrients, maybe a little bit with nitrogen, but maybe nothing with phosphorus that we could detect. But regardless, those algal treatment systems are effective at removing nitrogen and phosphorus, right? They removed 98% of the phosphorus and 92% of the nitrogen. That's huge. That's a very significant reduction of the amount of nitrogen in that wastewater. Second part of the experiment, when we looked at the anaerobic digestion and how much methane was yielded, we found that pretreatments and digestion with sewage sludge, both of those improve your methane production. But sewage sludge works a lot better than any kind of pretreatment. Doesn't matter if you heat it, add acid, any of that, it doesn't matter. What works best is just sewage sludge. So you don't need to do anything special. Take your algae, stick it in with your sewage sludge, digest them, and you're going to get way more gas than you would if you did any pretreatments. And if you take those results and plug them into the energetics model, we see that the best opportunity for you to have the most ecologically sound approach to the removal of this waste to create methane gas is to co-digest the algae with the sewage sludge at about a 90 to 10 ratio, somewhere in that range. 90 to 10, 80 to 20, something in that range, which is absolutely perfect, hits the sweet spot for a lot of wastewater treatment plants. So at the end of the day, the big take-home message here is that this works, right? This is a technology that's relatively simple, has no certain chemicals involved. If you couple an algal treatment system with a biodigester, you can improve the recycling by taking the carbon dioxide from the anaerobic digester, pumping that directly into the algal treatment system, and then you can take the algae from the algal treatment system, put it directly in with your sewage sludge, and you can improve the overall production of your methane gas. So this is a really big news story because it turns out that cost-wise, it's just as costly to make an algal treatment system and operate it as it is to create a chemical treatment system to remove those nutrients using chemicals that you have to mine and make and add and then remove. So cost-wise, which I didn't show you that stuff, it turns out that they're very, very equivalent. And so this is a technology that's out there and it should be widely used, though it's not widely used at this point. These are my literature cited, and I'll just end on this slide since it's more colorful and more interesting.